so there's a lot of theories on aging. I believe in all of them, but telomere shortening is the only one that gives us a really measurable limit to our lifespan that cannot be modified. We have when we do the math on the way telomeres shorten, and we understand that when telomeres get down to 5,000 bases, our cells lose the ability to function and we die of old age. But when we look at the rate that telomeres shorten, it gives us a theoretical maximum of 125 years that a human being can live. Nobody could live longer than that because of telomere shortening.
Hello, Dr. Andrews. So you've been central to the research around telomerase, including leading the team that discovered the human gene for telomerase in the 1990s. So you are now the CEO of Sierra Sciences, a company that you founded, which focuses on extending human lifespan through telomere maintenance. So welcome to Modern Healthspan, and thank you so much for joining us today.
You're welcome.
So Dr. Andrews, you've been looking at aging for a long time. So what led you to focus on telomeres in particular in the first place?
Well, as long as I've been studying aging my whole life, and all aspects of aging, I haven't really been just a telomere person. But I used to, even back in college and even high school, maybe in graduate school, I used to try to, as I started to learn how the mechanisms work inside cells, I started wondering, how could a cell know how old it is? And there had been data since 1961 showing that somehow when human cells divide, they know how many times they have divided and they know how many times they have left to divide. And how could a cell have a mechanism to count? And so I used to struggle with that. In fact, I started anti-aging clubs and things like that, and we used to have those kind of discussions. And one time I just came up with the idea that it's like there's got to be something inside of cells that's like ride tickets at an amusement park. Every time a cell divides, it loses a ticket. Because that was a mechanism that you could imagine actually functioning inside of a cell. There's a precedence for those kind of things and stuff. And so I started working on that, but I couldn't figure out, well, where are the ride tickets? And then one time I'm sitting at a conference, and that was in early 1990s, and I heard somebody talk about telomeres and the fact that they shorten every time a cell divides. And I suddenly realized these are the ride tickets. And before that person could even get off the stage, I was at the bottom of the stage asking him, has anybody figured out a way to add more ride tickets back or lengthen the telomeres? And he said, no, he's been working on it for years. He's been collaborating with labs all over the world, and nobody's figured it out. And now I already had a pretty successful career in biotech, and he knew who I was. And I said, well, let me come and work for you, and I'll have it figured out in 3 months. So 3 months later, I discovered my team. I led the team. There's no such thing as any one person making any discovery. But I led the team that discovered human telomerase. And that is why because we knew that telomeres couldn't get shorter in our reproductive cells, because if they did, our children would be born with shorter telomeres than we have. Their children would be born with shorter telomeres than they have. And after a few thousand years, we would have been not having any DNA left. So we would have been extinct as a species. So the reproductive cells had to have some way of preventing telomere shortening. And that's what I did. I started looking at reproductive cells to find out what's going on. And we discovered this enzyme called telomerase, which this telomere is still shortened. Every time a reproductive cell divides, this telomere is still shortened. Then telomerase comes in and re-lengthens it. So it's a constant back and forth, two-step process: shorten, lengthen, lengthen. Whereas all the rest of our cells in our body, we don't have this enzyme telomerase. We just have shortening. And so that's when I got that point, that's when I started figuring out, okay, let's figure out a way to make it so that all of our cells produce telomerase. so there's a lot of theories on aging. I believe in all of them, but telomere shortening is the only one that gives us a really measurable limit to our lifespan that cannot be modified. We have when we do the math on the way telomeres shorten, and we understand that when telomeres get down to 5,000 bases, our cells lose the ability to function and we die of old age. But when we look at the rate that telomeres shorten, it gives us a theoretical maximum of 125 years that a human being can live. Nobody could live longer than that because of telomere shortening. And it's the only thing like that in all the theories of aging, where you can actually say an absolute limit measurement and stuff. And but there's also things you can do to accelerate that shortening. That's almost anything related to a poor lifestyle choice. And that's why nobody ever lives to be 125. Because poor lifestyle choices and, let's say, poor genetics. Right. But that's what I've been doing. Ever since then, so that guy that was on the stage talking, I said, let me come and work with you. He was working at Geron Corporation at the time. And I went there and started working with him there. And after we discovered it, we not only showed that we could totally stop the aging process in human cells in a petri dish, which nobody had ever done before. We also could use what's called the antisense of telomerase to kill every cancer cell by causing the cancer to essentially die of accelerated aging. And the company, Geron Corporation, even though their name is short for gerontology, they decided they only wanted to focus on one, and they thought there'd be a quicker return on investment with focusing on cancer. So I started Sierra Sciences with their blessing to pursue the anti-aging. That's what I've been doing ever since.
Okay, a lot to unpack there. So if we think about telomeres, you said they shorten. So why do they shorten?
Well, shortening is actually the incorrect word. Okay, I'm just as guilty as everybody else. Telomeres, it's very rare for telomeres to actually shorten, and that's like lifestyle choices like smoking, things like that can cause accelerated telomere shortening. But telomeres actually don't shorten. What happens is that when a cell divides and it makes a new DNA, that new DNA is made shorter. Okay, so there's no degradation, there's no wear and tear, there's no unraveling, all the words you ever hear. And in shorten, I even use the word shorten, as I said. But what's really happening is just that new DNAs are made shorter. As a result, the average length in the cells, average length of telomeres in the cells gets shorter, but without being shortened, if that made sense.
Right. So you said that humans do not express telomerase in any normal cells, right? Only in, I guess,
reproductive cells.
Reproductive cells. So does any mammal express telomerase, or is it, does any animal, does any mammal?
Yeah, most do. Most, it's very few animals that don't. In fact, there's humans, most non-human primates, dogs, cats, horses, sheep, pig, and deer. Those are the only animals so far that have been shown to actually have the telomerase not produced in all their cells. So mice, for instance, they have telomerase produced in all their cells. They don't age by telomere shortening. That's a common misconception. There is some telomere-related biology underway there that's kind of unusual, but the telomeres in mice are so long that even and telomerase is turned on in all the cells that it really doesn't have any effect. But for some reason, when mice get really old, there's a sudden rapid shortening of the telomeres that's been published in some papers. I've actually never seen this, but that might be playing a role in the aging of mice. But mice mostly age from oxidative stress and mitochondrial dysfunction, not telomere shortening. And most animals. And then there's a lot of animals that don't even have the other like mice, they still age. They obviously have very devastating aging. But there's some animals that have telomerase produced in all their cells. One mammal in particular is the humpback whale. Humpback whales don't have any telomere shortening. They have telomerase produced in all their cells. They rarely ever get any diseases, and they have undetectable aging. Okay, people have found it's really hard to know how old a whale is because they don't have rings on a tree that you can count. So, but people have found like a harpoon in a whale one time, and they carbon dated the harpoon, and it was 130 years old. So the whale had to be older than 130. Now, there's other animals like that too. Lobsters, but they're not mammals. Okay, so lobsters have no detectable aging, never get sick. Let's see, tortoises never seem to have never seem to get sick and don't have any detectable aging. Some birds, some fish, clams are unique. Well, first, let me say that all these animals have undetectable aging, but nobody can really tell how old they are because nobody ever started measuring aging. And the only way to measure how old a lobster is to be there when it's born, have it in an aquarium, and just watch how long it lives. And nobody ever thought of doing anything like that until the time of Darwin. In fact, Darwin had a pet tortoise named Harriet that died something like 180 years old, and it wasn't from aging. Now, there is the one animal, clams. Clams do have rings on a tree. Every year they get a new stripe on their shell. And people have now counted them and found that there are clams over 500 years old. And it's all because they have telomerase produced in all their cells, they have no telomere shortening, and they rarely get sick from anything else, which is a suggestion that having long telomeres and keeping telomeres long is a very healthy thing to do. So,
Interesting.
we're unique.
Yeah. I saw that you talked about we evolved aging, right? Aging was evolved as a way to remove older people or older individuals so that the younger. So I thought that would be conserved, and so there would be telomeres would work the same in like our ancestors. But from what you just said, that's not true. So different animals age in different ways. So I guess, and so they would be not very good models for aging for humans anyway. But also it would be. So is there any animal which is a good model for aging for humans? It would be like dogs or monkeys.
I think the best animal for studying aging in humans is pygmy marmosets. Okay, they do have telomere shortening, and they're as small as mice, so which would make it so that preclinical studies would be easy to do. But there's actually only one smaller primate, and that's called Madame Bertha's mouse lemur. But we found out that lemurs don't have telomere shortening. They have telomerase produced in all their cells, and they're actually closer related to mice than they are to humans. And we didn't actually realize that until after we found that lemur cells do produce telomerase, and we started looking in detail at the evolutionary tree and found that the very first branch between humans and where humans and mice separate, that first branch on humans is lemurs, and I think they've just misplaced it. I think it should be over on the other branch with the rodents. But the second smallest primate in the world is the pygmy marmoset, and that's what when I speak at conferences, that's what I always say that I want to do my preclinical studies on, is pygmy marmosets.
Do humans express? So we have stem cells. We have like bone marrow stem cells, and so do they express telomerase, or is there also a limit to how many times they can divide?
No, stem cells do not produce telomerase. The exception is when you put stem cells into a petri dish and you start to let them divide really quickly, then you get very low levels of telomerase produced. But that's, I think, because every time there's a new DNA produced so when the cell divides, the DNA has to be duplicated. As soon as that new DNA is duplicated, for a few seconds, the gene is turned on because the gene repressor hasn't found the site to bind yet to turn the gene off. So I think it's leaky expression. Now, that said, human embryonic stem cells, which are really reproductive cells, they do have telomerase produced in all their cells, and they're only one cell, but they do produce telomerase, and so do induced pluripotent stem cells. Okay, so those two do produce telomerase, and believe me, we have done everything possible to try to find out why induced pluripotent stem cells produce telomerase, and before you induce them to be pluripotent, why they don't. And we have not been able to figure that out. And nobody else has either. You make induced pluripotent stem cells from the Yamanaka factors, and you can do 4 or 6, but we've tried using all combinations of 3, all combinations of 2, all combinations of 1, and the only way to get telomerase produced is to use all the Yamanaka factors. And which tells me that the rejuvenation that you see from induced pluripotent stem cells is actually more de-differentiation than rejuvenation. And the only rejuvenation that's actually occurring there, when you hear about partial rejuvenation, things like that, or partial reprogramming and stuff, is the fact that telomerase is being produced and lengthening telomeres. And that's the main cause of rejuvenation, even from partial reprogramming or genetic reprogramming.
Right. So I was going to ask about, so partial reprogramming with the Yamanaka factors does lengthen the telomeres, even if it's only partial, but it's still.
It actually keeps them from shortening. So it's a tie. Like I said, there's 2 steps, shortening and lengthening. It's like a tug of war, but it's a tie in the tug of war with induced pluripotent stem cells and human embryonic stem cells.
So there's, we have spoken to some people who have been using like exosomes or the secretome from embryonic stem cells or from young blood plasma. And this seems to have a positive effect on the organism generally. Do you know whether these factors would be able to extend telomeres, or do you think it's a different mechanism?
It's a totally different mechanisms, apples and oranges. Yeah, it's unrelated. But they produce like cytokines or other molecules that will have a positive effect on cells, the rest of the cells in the body.
But then you're still going to hit the same problem that you will run out of telomeres.
Yes.
Right.
And sometimes you actually might do it sooner, because if the exosomes or stem cells are doing anything that might induce cell division, as soon as that happens, you're actually accelerating your telomere shortening. And there's a lot of things that people do that actually do that, like human growth hormone and microdermabrasion and immune boosters. All of those things induce cell division, which only increases the rate of telomere shortening. But I always tell people to do them anyway, because what's the point of living a long time if you're not living? It's like
so is there any way to extend telomeres without telomerase? It does require telomerase.
No, there is one way, and it's called the alt pathway, alternate lengthening of telomeres, it stands for. And we only see it in cancer cells, really, some cancer cells, where a process of recombination, where just having the 2 DNAs, so 2 chromosomes recombining, actually can result in telomeres getting longer. I'm not aware of any case where anybody's ever demonstrated that in a non-cancer cell, but when telomeres get really critically short, all kinds of chromosomal rearrangements and mutations occur that cause things like telomerase to turn on or the alt pathway to turn on to keep telomeres long. But the cells have to become cancer first. They have to get critically short telomeres first before that can happen.
Time flies. It's almost the end of 2024, but it's never too late to prioritize your health. There's still plenty of time to make a positive transformation before the new year. Every little step counts. Did you know that eating healthier can lead to notable results in just 1 to 2 weeks? Regular workouts can make a significant difference in 8 to 12 weeks. And improving sleep quality can be felt in as little as 2 weeks. A basic nutrient like magnesium is crucial for overall health. I personally use Magnesium Breakthrough by BiOptimizers. More than just a supplement, Magnesium Breakthrough is a comprehensive formula containing 7 forms of highly bioavailable magnesium. It's scientifically designed to improve sleep quality, enhance workout recovery, and boost muscle growth. Imagine the progress you could make in the next 90 days. We challenge you to make the most of this time. Combine Magnesium Breakthrough with your health and fitness efforts and see how far you can go. To help you get started, BiOptimizers is offering a special 10% off Magnesium Breakthrough when you use the code MODERN10 at BiOptimizers/modern. Don't let another year pass without reaching your health goals. Okay, so the mechanism whereby we slow the telomere shortening. So the only way we can do that is basically to reduce the number of divisions. Right? There's no other way.
You can decrease the rate of telomere shortening by leading a healthy lifestyle. And for instance, vitamin D and supplements, vitamin D and omega-3 fatty acids have been well published to support that they reduce the rate of what I call accelerated telomere shortening. But there's all kinds of things like losing weight, quitting smoking, meditation, all these kind of things can decrease the rate of telomere shortening, but they don't lengthen the telomeres, okay?
Yes, but the underlying mechanism must be that they reduce the number of cell divisions.
No, they reduce them like free radicals, okay? Free radicals can actually clip the DNA, okay? So when I said DNAs don't shorten, they don't degrade and stuff like that, I didn't say they don't. I said it was rare, but you can get that by especially smoking. Smoking is a terrible thing for causing accelerated telomere shortening by inducing free radicals that will then bind to the telomeres and actually clip them. And so that's one way so the telomere is shortened by other mechanisms besides cell division. But cell division is the main way for a healthy person.
We have multiple different tissues which divide at different rates. When we say telomere length, I mean, what are we measuring? Because I would think different cells and different tissues would have different histories, and so they would have different lengths of telomeres.
Exactly, yes. Even when a person dies at 110 years old, there are cells in that person's body, tissues in that person's body that still has long telomeres. So it's a function of how many times has a cell divided. Now, there's cells can be divided into different groups. There's consistent replicators, partial replicators, and non-replicators. Okay, and a nerve cell would be a non-replicator, for instance. But so the consistent replicators, they're the ones that are always dividing, and that includes the cells lining our gut, cells lining our immune system, and our skin. Okay, so those cells are always dividing, and the immune system, so the blood cells, that would include the blood cells. So those 3 things are probably the things that correlate best with aging and telomere length. And it's those 3 things that, when they fail, is one of the major causes of death by aging. So you can measure the telomere lengths of skin and cells lining the gut and cells in your blood cells, and find that actually those shorten at a consistent rate. Whereas other tissues, like neurons and stuff like that, as soon as they differentiate into a neuron, they don't have any more telomere shortening. Their telomeres just remain long.
Right. So it's the one that runs out first that's going to be the issue. Right. So can you use measuring telomeres to estimate biological age? And is measuring telomere length a good way to measure biological age?
It would be if we had good ways of measuring them. Now, I've invented most of the procedures that are used now to measure telomere lengths, but they're very difficult to do and they're very inaccurate. And it's partially because even a single cell has telomeres of multiple lengths. So you don't get a single length, you get a smear of values. So I do have some protocols that I use here that are very accurate at measuring telomeres, but they're just so expensive to do, they could never be used for personalized medicine. And hopefully we can figure out a inexpensive way to do it. But yeah, there are companies out there. There is one company that I think does a really good job, and that's a company called LifeLength. But it's difficult to do, but they do give you an accurate answer. But I think we can even be more accurate than them using the right tools. But it's expensive. Now, since I believe, and we have data that suggests that we haven't published, that DNA methylation changes in DNA are actually controlled by the telomeres. So when we take cells and we have inducible telomeres where we can turn it on and off at will, we can make telomeres long in a cell, then we can make them short in a cell, and then we can make them long again. And when we do that, we see the DNA methylation patterns change back and forth proportional to the telomeres. So I always say, I think the best way to measure aging is not telomeres, but only because it's so difficult to measure them. I think DNA methylation and IgG glycosylation are the two best ways to measure aging right now.
Okay, so the IgG glycosylation is the glycan age?
Yes, glycan. Yeah, I actually, if I had to choose between the two, I would go with glycan age. I think glycan age is the best measure of aging that exists right now, besides what I call the Betty White test, which is when I'll show a picture of Betty White when she's 25 and 95, and I'll say, anybody here not know which photo of Betty White was taken first? But we don't know what it is. I call it qualitatively quantitative. Okay, but when a patient goes into a doctor's office and says, "Oh, I wish there was something that would make me young again," they're not saying, "Oh, I want my telomeres to be longer, or my DNA methylation patterns to change, or my Ig glycosylation patterns to change." They want to pass what I call the Betty White test. They want to look and feel and behave 25 again. And a lot of people are claiming to have cured aging just by reversing the biomarkers, but the biomarkers actually didn't reverse aging. They just reversed the biomarkers.
Right. And it would seem like from what you said before that if you reverse the biomarker, you reverse the epigenetic age. That should be related to the telomere length. But yeah, I'm just trying to think how that will work. So we have had some protocols which reduce the epigenetic age. Does that impact the telomere length at all?
Well, no, it doesn't, but vice versa is true. The telomere length does affect the epigenetic age, but you're talking about epigenetic mechanisms, okay, which is what DNA methylation is. That's not epigenetics, okay? That's just a misnomer, okay? People call it epigenetics, but it's actually just an epigenetic mechanism because it's known that methylation will change the gene expression. The only really good way to do epigenetic measurements is total RNA sequencing, and take cells and just sequence all the RNAs and then just look at what's what and how many of what genes are turned on at high levels, what genes are turned off by measuring the RNAs. And that's just so expensive, nobody does that. But yeah, but that's something that, again, we do here. So when we measure epigenetics, we don't really look at DNA methylation. We do total RNA sequencing with a company called CD Genomics. We actually send the samples out to have CD Genomics do total RNA sequencing for us.
Okay. So we did talk a little bit about how we can slow our telomere shortening. So if we can dive into that a bit more detail. So it's based on, I guess, the replication of the fastest replicating tissue that we have. So what kind of lifestyle choices can we make to reduce the telomere shortening, and how much difference do they make?
Well, let me first, you were talking about like the cells, the tissue with the fastest shortening of telomeres. Now, so there's a lot of genetic and lifestyle choices that will target particular tissues. Like, for instance, if you have muscular dystrophy, you have a mutation in the dystrophin gene, that will cause muscle cells to die quicker. And as a result, other cells need to divide to replace those killed cells. And when those cells divide to replace the killed cells, the telomeres get shorter. And now, because that replacement of dead cells is going on at a faster rate, the muscles will die of old age long before the other tissues of the body. And that's why people die of muscular dystrophy. I can use the same kind of stories for kidney disease and Alzheimer's and liver cirrhosis. Liver, like drinking alcohol, that's something that kills liver cells. Okay, and that's okay when you're young, because when you kill the liver cells, other cells in the liver divide to replace those liver cells. But when you get into your 50s and you've been drinking your whole life, your cell, you've had so much cell division that now the cells that would normally replace the killed liver cells can no longer do it because their telomeres are too short. And so that's when you get liver cirrhosis, and you end up dying of liver disease. And now, because I went on that tangent, I forgot the question. What was the question again?
Oh, dear. So what lifestyle choices? Yeah, for
just everything we've ever learned as we were growing up practically is a good way to keep telomeres long. But don't drink alcohol, don't smoke, lose weight. Meditation is a great way of decreasing stress. Anything that causes psychological stress will accelerate telomere shortening. There's actually some studies now showing that pessimism actually causes accelerated telomere shortening. So being more optimistic actually lengthens telomeres. And if there was only one study that showed that, I would say, well, I'd got to see that data reproduced. But there's been 2 studies now showing that people that are pessimistic have shorter telomeres than people that are optimistic. So if you don't think you're going to live to be 100, you're probably right. More likely right than somebody who thinks they'll live to be 100. But what else? There's so many things that we do to ourselves that cause accelerated telomere shortening. But like UV exposure, it's just about everything that. Hold on, because I gave a presentation just a few days ago. I have a list. So, like improper exercise. Okay, so if you're consistent in your exercise, your body doesn't think that exercise is a threat to you. Okay, so it does not induce an inflammation response. But if you are an occasional exerciser, even if you're a good marathon runner, but you only train once a week or once every 2 weeks, you run that marathon, you're going to induce a lot of inflammation, and inflammation causes accelerated telomere shortening. But if you're a consistent exerciser, you can run a marathon and get no inflammation. And so proper exercise, I always say when I speak at running groups or sporting functions, I'll say, keep it fun. When it quits being fun, quit. Okay, and come back and do it another day, because if you do that, you'll find that eventually you start getting really good and you can start running 100 miles, in ultra marathons, and I've done many 100-mile races where before, during, and after I got blood samples taken to measure my CRP and other inflammatory markers, and I had very, very little induction of inflammation. That's because I run every day, I'm actually, I think I'm something like, I haven't missed a day of running in over 4 years, and there's some place that keeps records of that, and I'm something like in 2,500th place in the world overall for most consecutive days running ever. But it's people think, well, God, you run every day, that's doing your body harm, but it's the exact opposite. Even with knee joints and things like that, it's the occasional exercise that causes knee joints. Running every day keeps your joints lubricated, and you never have knee joints. I have a perfect control sample, okay, because I have an identical twin brother who's from like 25 years ago. He and I, when we look at photos, we can't even tell each other apart. We don't know who's who. But he doesn't run like I do. He doesn't exercise like I do, and he is suffering from knee joint pain and hip pain and ankle pain, things like that. And he's talking about getting his knees replaced, etc. And he probably will really soon. And everybody thinks, well, I'm the one that should be getting my knees replaced, not him. But that's because, as I said, it's when you're occasional exerciser, and then exercise that induces inflammation, and then inflammation induces damage to your joints. I'm trying to think of what else to mention. I know what I want to mention. A lot of foods, everybody's different. A lot of foods cause inflammation. And there are two really good tests that people can do. One is called ALCAT, ALCAT, and the other one's called MRT. They're both food sensitivity tests. And those will tell you what foods cause inflammation in you, and therefore all you got to do is quit eating those foods and eat the foods that don't cause inflammation. And that's another way of decreasing the rate of telomere shortening.
Just going back to exercise for a second, because I do remember there was a discussion around, running marathons was not good for you because it put stress on your body for an extended period of time that your body was really not designed for. So what you're saying is that is incorrect, that if you are properly trained and you've spent the time building up, then even running marathons, because you do ultra marathons, right? So it's 100 miles. So that's like 24 hours. How long does it take to get 100 miles?
I've broken 24 hours on a few occasions. But yeah, the thing isn't, don't struggle, keep it fun. Okay. And as I said before, so even if you are, as you said, well-trained and stuff like that, you can still cause inflammation if you push it really hard. And so if it's unhealthy, it's unhealthy. So when you push it, if you're one of these people that cross finish lines and get on your hands and knees and throw up, you've done your body a great injustice. Try to keep it fun. That's all I say. It's just keep exercise fun, keep competition fun, even if you don't win the race. There's other things to win. It's like you can. Like I used to hold the world record for the most 100-mile races run in 1 year. Okay. Now I'm proud of that, but I never won a race. Okay. And so there's other things, other goals to seek that would be healthier.
Interesting.
Yeah.
Yes. Okay. So are there any supplements that would help? We talked about vitamin D and
omega-3 fatty acids.
Omega-3 fatty acids. Yeah. Anything else?
Every single antioxidant and every single anti-inflammatory is going to decrease the rate of telomere shortening. So now, like, and I would refer people to the Kaufman Protocol, a book called The Kaufman Protocol, written by Dr. Sandra Kaufman. That book is probably the best book, not probably, it is the best book ever written on what supplements to take and how much to take and when, and who should take what. And it's all in formulas. It's figuring out what's the right thing to do. But I used to spend time talking on stage and talking about supplements and stuff like that. And so I read Dr. Kaufman's book. And then now I just tell everybody, read Dr. Kaufman's book, because I've never seen anybody get it more right than she has. But yeah, there's a lot of supplements. And I do take, for instance, I do take more than one anti-inflammatory, and I do take more than one antioxidant, because different antioxidants and different anti-inflammatories have effect on different parts of the body. So,
right.
And
yeah,
don't ask me to remember what those are, because ever since Andy's written her book, I've just kind of focused my attention on other things now.
Okay. Yes, and she's written another book now.
Yes. So you know her? Oh, good.
Yeah.
She's I think those are the best books ever written, both of them.
Excellent. Yes, we spoke to her.
Okay.
What are you studying at Sierra Sciences in terms of ways to extend telomeres?
The main thing we're doing is we have developed a high throughput robotic screening protocol where we can test up to 4,000 chemicals or nutraceuticals or peptides or anything a day. And so this assay with the robots just looks at the production of turning on of the telomerase gene. So so the main thing we're doing, we don't know what the best thing is. The advantage of looking at chemicals, pharmaceuticals, is that when we find chemicals that work, and we have found over almost close to 1,000 different chemicals now that will induce telomerase. But because they're chemicals, we can engineer them, we can modify them, we can do what we have medicinal chemists that work here that will redesign the molecule to increase activity and decrease toxicity. Now, I don't want to make it sound like all chemicals are toxic, because turns out more nutraceuticals are toxic than the chemical. We have a lot of nutraceuticals that are toxic too. But the nutraceuticals, and we have a lot of nutraceuticals that we've discovered that works. And, something that people should be aware of is that we have discovered the best. In fact, every nutraceutical on the market was pretty much invented partially or totally by me. Okay, so I really understand all this stuff, but we have now discovered some of the best the best nutraceuticals ever for turning the telomerase gene on, and they should be marketed by a company called Touchstone Essentials, beginning in February of 2025. So they're actually in production right now. Put in the different, because it's going to be a mixture of strong nutraceuticals that induce telomerase. They're going to be mixed together, put into capsules, and they're going to start selling them in 2025. Now, I don't do any marketing, even though it sounds like I just marketed. I'm just excited about the fact that these things really work in our hands in our assays and stuff like that. And I'm glad to see that they're getting out of there. But we don't market them. I license them to some other company, Touchstone Essentials, and they are now preparing to market them. But so the nutraceuticals, they can, well, when we discover a nutraceutical, we cannot modify it like we can chemicals to make it better, because as soon as you modify a nutraceutical, it becomes a pharmaceutical, and then you have to get FDA approval. But we do have pharmaceuticals, chemicals that we are trying to get turned into pharmaceuticals that could be on the market in 10 years or so, depending on how long FDA clinical studies take. But I'm going to be honest with you, the big problem isn't how long it takes. The problem is the funding, finding people to fund these clinical studies when we already have things ready to go is really the big obstacle. And again, my company doesn't do testing. We would license these to other companies, and we have, and they're having trouble finding the funding stuff to do the clinical studies. Gene therapy is actually the best, is the best thing right now available to lengthen telomeres. And a company called BioViva is actually now providing these. Have you interviewed Liz Parrish yet?
We did a while ago. Probably time
Okay.
to talk to her again. But
Yes. Yeah, what she's doing in her company is very amazing. She's now providing gene therapies, and we're working with her in trying to improve the safety of the gene therapies and get more testing done to find out how they work and find the right dosing and stuff like that. But in the meantime, she's already providing it at doses that are safe and efficacious, but not as efficacious as what I want. You know, I want to pass what I call the Betty White test. I want to look and feel and behave 25 again. And so far, nobody's willing to try the gene therapies at those doses because there's a very high chance that it won't be very healthy for the person. Like when we've done the gene therapy studies with animals, and I said before that mice don't age by telomere shortening, but we did provide the gene therapies to some other group that treated mice with them, but they first engineered the mice. So they created abnormal mice that actually do age like humans by telomere shortening. And when they treated the mice with high dose gene therapies, they saw complete reversal of aging. And this was on Diane Sawyer. Diane Sawyer did a interview about that. If you haven't seen that report, the doctor was Dr. Ron DePino. Now the mice still had their other aging mechanisms, such as the oxidative stress and mitochondria dysfunction. So they didn't become immortal, but they definitely had a reversal of aging that occurred as a result of high doses of gene therapy. But I got to say that the gene therapy, the problem with gene therapy is not the telomerase. It's actually the delivery mechanism. Gene therapy is really a way of delivering a new gene to your cells that can do whatever. In this case, the gene therapy is delivering the telomerase gene to cells. And we have engineered the telomerase gene to be very strong. So the delivering the gene to the cell is actually what's toxic, okay? And that's what's got to be worked out. So when you do gene therapy with mice, not all the mice survive the gene therapy, but the ones that do then show signs of age reversal. Now we don't want to start treating people with gene therapies at those doses because we don't want to find that 10 to 20% of the people die from delivering the gene, but the remaining 80% get younger. Okay, that's not going to be anything that's going to be very satisfying. So we have to work on finding safer ways of stuff to do the gene therapy before we can really get to the high doses. But I think people should keep an eye on BioViva because when we actually do have all that stuff figured out, it will be BioViva that will be marketing those companies because we're very big fans of the ethics. Let's say a lot of companies will fake data, they will make false claims and things like that. But I like working with BioViva because they never do that.
Okay, going back to a couple of things in there. So one of them is, how is telomerase suppressed in humans? Why is it never expressed?
Nobody knows yet. Okay, and this is very frustrating. And the fact that so much work and so many publications have come out on people trying to figure that out, and nobody has actually figured it out, tells me that the telomerase gene is actually repressed by multiple mechanisms. Okay, so I kind of think of it as when I talk at conferences and stuff like that, I'll talk about how next to the gene is a promoter, and a repressor binds to that promoter, repressor protein binds to that promoter and turns the gene off. But I think there's actually like multiple repressor sites in the DNA of the promoter, and each site has a different repressor binding to it. And so, I said we have found 900 plus almost 1,000 different drugs, chemicals that will turn on telomerase. We think that some are de-repressing one repressor, some are de-repressing a different repressor. And we're very excited about doing what we call the synergy experiment, where we start combining different chemicals, but this is extremely expensive. And the thing that's holding us back is the funding. And that's just extremely frustrating, because it literally costs us $2 million a month to do the robotic high throughput screening to test different drugs. And it's just, especially since COVID, it's been very hard to get keep that funding going. But I think if we could get the funding that we need, and I'm talking about funding that's way beyond angel investors and things like that. I estimate that for us to actually find a chemical or a mixture of chemicals that will actually turn on telomerase enough to reverse aging will cost about $130 million and take about 3 years to do. And that seems like, well, any billionaire who's suffering from age-related diseases would just jump in and provide that funding, but they don't. All the well-known ones always are wanting to know how quick can they get a return on their investments. Even though they already have $1 billion, they want us to do other research to come up with quicker returns on investment. And so my focus lately has been on the billionaires and stuff like that, multi-billionaires that nobody knows because they just keep their life secret and trying to get their interest. And just the hardest part about that has been being able to reach out to them. But I am talking to a few now to see if we can get that stuff going again. But I do believe that as soon as somebody provides us with $130 million, we can get a pill that people can take that will reverse, well, let's say, will make telomeres longer and in all the cells of the body. It'd be the first time that's ever happened in a human. And when that happens, I'm not going to say that's going to reverse aging. What I'm going to say is we then get to look to see what happens, okay? Because lengthening telomeres as a way of reversing aging is still a theory. I mean, it does work in mice, it does work on human skin grown on the back of mice, and it does work on human cells in a petri dish. But I can't say that it actually will work in humans until we actually do it and test it, okay, and see what happens. And so I have written clinical protocols to do it, and the clinical protocols essentially would cost my company $1 million, or whoever licensed it from us, it would cost them or us $1 million to test one person once, okay, just because of all the controls that would be needed. And because if it doesn't work, we want to know why it didn't work so that we can then fix the problem and make it work. So it's expensive, but I don't know how I got on the subject, but I do see a lot of hope for both gene therapy and chemicals. Oh, it's because you're asking me about what we're doing now. And yeah, and we do have our strongest chemical right now produces about 16% as much telomerase as we need to stop the aging process, or stop the telomere shortening, which theoretically will stop the aging process. And because we've screened over 300,000 different chemicals, I think it's more like 350,000 different chemicals, and found over 900 different chemicals that work, that's enough data for us to extrapolate to show how much more research we need to get to the point of having a drug that's going to be potent enough to stop telomere shortening. And That's where I get the 3 years from. So it's actually a range of 1 to 3 years and $130 million more is what we need. Because we've already spent like 30, close to $60 million to get to where we are now. And we just need another 130 million to finish the research. And then at the same time, we do want to promote the gene therapy. I think the gene therapy will come along quicker even than that. So the work we're collaborating with BioViva, I think will produce some exciting results pretty soon. And there's a lot of companies that do gene therapy, and a lot of companies that are doing gene therapy with telomerase, but they all have the problems, and really none of them have the expertise to actually really know how to do the gene therapy to make it work. Whereas I've been doing gene therapy since the 1970s. I've designed practically every kind of gene therapy you can imagine, stuff like that. So I think that given the proper amount of funding, we can get gene therapy and then small molecule chemicals to be able to stop telomere shortening and hopefully, as a result, reverse aging.
Okay, so going to the gene therapy for a second. So in the gene therapy so first of all, the gene therapy offered by BioViva right now is something that, well, actually, they don't offer it. I think they kind of manage it, and it's delivered by another party.
IHS. It's a company called IHS that they get most of their business from Liz Parrish is promoting them. But yeah, Liz Parrish analyzes the data, but they're essentially working for her, for BioViva and doing.
Right, but it's not in the US, it's somewhere else.
Yeah, it's right now, gene therapy is considered a pharmaceutical type therapy and therefore requires FDA approval to do it correctly. And so to get it done, people need to go outside the country to do that right now.
So Liz Parrish has had telomerase gene added.
I gave it to her in this very room that I'm standing in right now. I gave her the gene therapy back in 2014 or 2015. And I'm embarrassed to say that I told her, don't tell anybody where you got it, because I didn't really approve of her treating herself with it, but she was pretty insistent on doing it. And so she found another lab to scale it up, and then another lab to actually treat her. In hindsight, I wish I had been more involved, and because I think I could have managed the results, interpretation of the results better. Because as soon as I heard that she had done it, I started asking a lot of questions, and turned out nobody thought of trying to answer the questions until I asked them, which was too late. But yeah, so she's treated herself. She's done it more than once now. And she's also treated herself with other gene therapies. There's so many rumors going around, just hearsay, false things about gene therapies that aren't true. And one of them is that you can't do it more than once because of immune responses, but that's not true whatsoever.
Yes. Do you know how she's doing at the moment? I mean, it's what, 10 years later?
Yeah. Okay, so I can only be anecdotal. Okay, when she first treated herself, I was at an A4M conference, and she was coming from the treatment to the A4M conference. And I told her, I said, okay, walk into a room and I'll have my eyes closed, and then tell me when you're there. And then I'm just going to open my eyes and I'm going to tell her, do I think she got younger or not? And I opened my eyes and I said, no, you did not get younger. Okay. But now 10 years later, there's no question in my mind that she has gotten younger. Okay. She definitely hasn't gotten any older. Okay. She looks younger now to me than when I first met her over 10 years ago. But as a scientist, I can't say that proves it works. Okay. Because not the right measurements. Well, I can't say that not the right measurements were done. I can say I didn't get my chance to measure things, okay? And I pretty much, it's going to sound like a kid who takes the football home because they can't be the quarterback. I don't believe any data unless I can do it myself, unless I reproduce it myself. And that's something that I would love to do. I just haven't had a chance to do it. Again, it's very expensive to do the testing. It's and you know, of course, she has tested the biomarkers like DNA methylation and stuff like that. And yes, she did see reversal and stuff like that. But it's those tests are, I mean, what does it mean? Did you reverse aging or did you just reverse the biomarkers? Okay, those are questions that still are unanswered and stuff like that. But there are those people that believe that reversal of biomarkers, reversal does reverse aging, then go sign up to get treated by Liz Parrish because it works. It works for that.
Right. Yeah, maybe you are correct. We should talk to her again.
She and I are working on a brand new type of vector for gene therapy. The vector is the part that delivers the gene that I think is going to be a lot safer and a lot more accommodating to all kinds of particular gene therapies. But I'm not at liberty, I don't think, to discuss what that is. But I'm pretty excited about it.
Interesting. Yeah, I think what she's doing is wonderful. I mean, like, very brave as well as taking and then making her data open and at least providing an N of 1 for more, forward-looking therapies. One more detail on the gene therapy. So when the gene is coming in on a virus, right? AAV.
Yeah, you can do a virus, you can do a non-viral thing. They're pretty much exactly the same. It's a coat, like a ball that you put the DNA in the middle of. What that ball is made of is really, there's 100 different things you can make it of. Anything that will get into a cell. So some are viral, some are non-viral, but they're really much the same thing. Sorry if I interrupted you there. I just wanted to.
Okay, no, that's interesting because I was thinking about, so the DNA does not, it must get into the nucleus, but it does not integrate into the chromosome. So it's like a separate piece of DNA.
Well, all DNAs are going to integrate at some rate. Okay. I mean, as I said, I've been doing gene therapy since the 1970s, and I've been just taking naked DNA and put it into a cell, and I can find cells that it did integrate, but it's extremely rare. I mean, it's so rare that I would have to do a lot of work to find one where it did integrate, but it does happen at a low rate. But with something like AAV, the rate is about as low as you can get. Extremely rare to have an AAV integrate into the chromosome. Now, lentiviruses essentially require integration in order for them to work, but their infectivity rate, meaning like how good are they at infecting cells, is almost perfect. Okay, so viruses, when you look at viruses, you use a score called multiplicity of infection. Okay, so how many viruses does it take to get 50% of the cells infected? And with lentivirus, it takes one virus per cell. Okay, you can get 100% of the cells, at least 50 to 100% of the cells infected just by using using no more viruses than you have cells. That's one of the reasons why we use that in our research. Adeno-associated virus, AAV, we have to use something like 300 to 3,000 viruses per cell. So it's got a low infectivity rate, but at least it doesn't integrate. And the problem with integration is that random integration of the DNA into the chromosome can cause cancer by turning on oncogenes or turning off tumor suppressor genes, by just mutating them, essentially because when you integrate a DNA into the chromosome, you've essentially mutated that DNA, that gene that you just integrated into. So and this, I'm just going to go on a tangent here. This really frustrates me about the FDA and stuff. And that's something that Liz Parrish and I do a lot. Is we've published a paper on something called best choice medicine to try to revamp the way the FDA works, because the FDA, like 20 years ago, there was gene therapy being done to treat the bubble boy disease. And anybody that had half a knowledge of gene therapy and how these things work knew that these kids were going to die of cancer because they were using a virus that integrates into the chromosome. And and I was horrified by the fact that they were even considering doing the study, and the FDA approved it. And the study was done, and sure enough, 5 out of the 7 kids, something like that, died of cancer. And it makes me feel like, boy, the burden of safety has to fall more on the principal investigator than the FDA, just because I don't think the FDA is always qualified to be able to decide what's safe and not. And sometimes you got to go with the scientific experts in the field to really figure that out. But that's just 20 years later, I'm still bitter about the fact that that was done. But yeah, so you don't want to integrate when you're doing a gene therapy on a human, unless you take cells from the body, like stem cells, and then treat them with a gene therapy that integrates, and then test those cells or sequence them to find out where did the gene therapy integrate. Did it cause cancer or not? And then find one that integrated into a safe place, then grow a lot of those cells and inject those cells back into the person. That's actually done nowadays, but it's still pretty much only done by the viruses that don't integrate, or non-viral gene therapies. And people say that non-viral gene therapies don't integrate yet, but they do, and they also induce immune responses. It's just marketing non-viral gene therapies is sometimes making false claims.
The DNA gets into the nucleus, and hopefully it doesn't integrate. We don't want it to integrate. So when the cell replicates, does it replicate your DNA as well? And will that DNA hang around for a long time?
No, it won't. And in the case of telomerase, you don't need it to. Okay. So let's say adeno-associated virus. So let's say 100 adeno-associated viruses get into a cell. Okay, well, when that cell divides, each daughter cell is going to have 50 adeno-associated viruses. And when those cells divide, each is going to have 25. And so after a few cell divisions, there's not going to be any virus left in the cell at all. But that's okay, because the goal is to lengthen the telomeres. Okay, and so once the adeno-associated viruses get into the cell, produce telomerase and lengthen the telomeres, you don't need the virus anymore. Okay, maybe 10 years from now, when your telomere, let's say you're a 40-year-old and you want to be 30 years old. So you get treated with the adeno-associated telomerase gene therapy, and that lengthens your telomeres to a 30-year-old. Okay, well, 10 years from then, you'll be 40 again. Okay, and you just get treated again, and you're back to 30. And so so it's no big deal that the DNA gets lost when you're doing gene therapy that doesn't integrate. But in some cases, it's not going to be good. Some cases you want to keep that virus around producing the protein on a consistent basis, because it's something that's needed in some biochemical pathway to make it safe. And so there's going to be cases where people need to be treated with gene therapies like weekly or monthly, just to keep healthy.
Right. Yes, that makes sense. And yeah, interesting. So once every number of years, sure, we don't know how many right now. Okay, so one last kind of topic of discussion. Telomerase and cancer. So cancer cells express telomerase, which is how they manage to divide endlessly. Otherwise, they wouldn't be able to. Does having telomerase in your cells increase the risk of cancer?
It should. I mean, if you were only to put telomerase into particular cells, it would increase the risk of cancer slightly, but it's not going to cause cancer. Okay, the cell still needs to have mutations that disrupt its division. It's causes the cells to divide. Okay, so cells are, there's what I call cell division control. Okay, so there's control features inside of a cell that tell a cell when to divide and when not to divide. In order for a cell to become cancer, you have to mutate those genes that control cell division, and then cell division gets out of control. Okay, telomerase isn't going to do that. Telomerase is just going to allow a cell to divide more times when it's supposed to. Now, the real problem comes in when telomeres get really short, then mutation rates skyrocket, chromosome rearrangements skyrocket, which increases the chances that you're going to turn on those oncogenes and turn off those tumor suppressor genes, and the cell's going to start going out of control. And then because of the high mutation rates, so now all of a sudden it becomes a cancer cell. Lack of growth control is what I call for cancer. Now that it doesn't have growth control, it'll start dividing further, and the telomeres will get even shorter. And then the mutation rates now are skyrocketing so much that it actually will cause a mutation that turns on the telomerase gene or turns on the alt pathway, like I discussed before. Then that cancer cell becomes immortal. Okay, now, if you treat the whole body with telomerase, you're also going to be lengthening the telomeres in your immune cells. So your immune system is your best defense against cancer. So if you are lengthening the telomeres in your immune cells, you increase the chances that if you did get cancer, your body would fight the cancer and kill the cancer. And what else was I going to say? So we have the immune system getting better, and we also have, by lengthening telomeres, the mutation rates caused by short telomeres decreases. So you have a less chance of getting a cancer. So it's kind of like a scale. I mean, it's like what's more likely to give you cancer? No telomerase or telomerase? And all the data right now is pointing to the direction that it's the lack of telomerase that's more likely to cause cancer than telomerase. That's why we get cancer so much already. Okay, so by producing telomerase in your cells, you're actually going to decrease the risk of cancer. Now, a lot of people completely misunderstand that, and I hope I explained it well enough so you understand what I just said. But it's like very few people, in fact, I teach continuing medical education all the time. I do teach continuing medical education on cancer. I was National Inventor of the Year for my cancer research in the United States one year. So I understand cancer really well. But I'm just amazed at how many doctors in the cancer field that I find that don't understand what cancer is and stuff like that. So there's a lot of confusion out there. People believe that telomerase will cause cancer. And I've gone through excruciating detail to go through that subject. Unless you want to spend another hour talking, I can do it right now. But I've gone into excruciating details in some videos. I gave a talk in Tokyo in 2017, where I spent almost an hour proving that telomerase doesn't cause cancer. It decreases the risk of cancer. And that video is on my website. People can go to that website. I've sent so many people there. I remember telling everybody, start watching at 25 minutes and 25 seconds, because that's when I start the subject. But I not only go into the details about why telomerase doesn't cause cancer, I also go into the details of like why some people believe it does and who's spreading the rumors that it does when it's not true. Yeah, so there's, if some people go to my website and go to my videos, just look for my 2017 presentation in Tokyo. And that's probably the best place to, best summary ever given anywhere on why telomerase doesn't cause cancer. And the reason why I did that, incidentally, is because this was a high publicity presentation that I was speaking at in Tokyo. When I landed by plane and was taking a taxi to the hotel, I was shocked to see billboards with my picture on it about this presentation that I was going to be giving about telomeres and telomerase. But people got on the radio and started announcing that, telomerase causes cancer, trying to discredit my presentation. So I ended up staying up all night and put together everything I know about cancer and telomeres and stuff like that, and included that into my talk. And you'll hear it 25 minutes and 25 seconds. I say, okay, I'm going to stop my talk now and switch subjects, because I said, how many people here heard the news last night about telomerase causing cancer? And half the room raised their hand. So I said, okay, I'm going to tell you now why that's not true. And at the end of the talk, I asked how many people still believe that telomerase causes cancer, and not a single person raised their hand after everything I showed, data and everything like that. And I put it, I thought it turned to be one of the best presentations I ever gave, even though I did it without hardly any sleep at all. But yeah, so there's rumors out there that telomerase causes cancer. It's very rare to hear that nowadays, because I think most people now understand that it does not cause cancer. And people on short telomeres cause cancer. Lack of telomeres causes cancer. Long telomeres help your body fight cancer and help prevent cancer by decreasing mutation rates. And high levels of telomerase does the same thing. And that's why, when I was talking about lobsters and humpback whales and clams and stuff like that, they never get cancer. They have telomerase produced in all their cells, and they never get cancer. Okay. Now, mice get cancers at extremely high rates, because mice have so many other problems. I would hate to be a mouse. Okay. Their biology is so bad. And, you'd mentioned something about evolution. I do talk a lot. My PhD, one of my PhDs essentially is in evolution. It's called population genetics. It's what's the how and why of evolution. And I do talk a lot at conferences about why we evolved an aging process. And people will say, well, how come some animals didn't evolve an aging process? And I say, well, they haven't evolved it yet. Maybe a million years from now they will. Because evolution is just, it turns out to be that I wasn't going to get into this later, but just quickly, I'll just say, adding diversity within a species increases the ability of that species to have at least some members in the species survive rapidly changing environments. So if you have a lot of diversity within a species, you have a higher chance that the species will not go extinct as a result of a rapidly changing environment. Well, getting rid of the longer lived, so after you've raised your young, getting out of the way increases the opportunities for the offspring to interbreed, which increases diversity. Okay, because when the offspring interbreed, there's more diversity that results from just letting the parents rebreed. So it turns out most species have evolved some way of eliminating the longer lived, just for the purpose, because it's not a purpose, because evolution isn't a force. Evolution is just a result. But as a result, those species that ended up accidentally evolving a way that eliminated the longer lived, those species had a better chance of surviving rapidly changing environments. And that's why most species now have an aging process.
Yes, I saw that. I thought that was really interesting way of thinking about the reason that we age. And it sounds like it would be almost programmed. It's like a program.
It's not a theory. Not a theory. It's a fact. I mean, no question about the fact that in higher diversity gives the species a better chance of surviving, and eliminating the longer lived increases diversity. So that is happening. Whether or not something else is also happening, those would be theories. But the fact that it's the survival of the species is higher by eliminating the longer lived, that's not a theory, that's a fact. But
yeah, that makes sense. Okay, one last detailed question. Does the cell have a way of detecting that the telomeres are short? Or is it just that the short telomeres, when telomeres become too short, things start to go wrong, right? So a cell can become senescent when the telomeres become too short. Is that like an active process, or it's just the telomeres become too short and things go wrong?
There are biochemical pathways that have been discovered that happen as a result of short telomeres that induces the cell to become senescent. Okay, I personally don't think the whole story is understood yet. I think it's going to be more complicated than is published so far. But the fact is that when a telomere gets down to about 5,000 bases, it induces a lot of changes in the cell that causes the cell to go into senescence. And then a few weeks after going into senescence, the cell dies. And but even long before the telomere gets what's called critically short, there are changes in gene expression that affect aging. Okay, so like you can tell a person that's 40 looks older than a person that's 30, even though their telomeres are not critically short. Okay, but it's because as telomeres get shorter, they affect the genes on the chromosomes. Now, nobody has figured out exactly how, but because my background is so much into gene expression and stuff like that, I've been studying it since the 1970s. I've noticed that there's a tremendous similarity between telomeres and something called enhancer sequences. And enhancer sequences are things that can be like 10,000 bases away from a gene and affect the expression or the epigenetics of that gene. And it works by the DNA folding over, bringing the enhancer over to the promoter. And then proteins that are binding to both interact with each other and turn the gene on or off. So the enhancer can be a long ways away, but the folding of the DNA brings it over. Well, I believe that's what's happening with telomeres. Telomeres are remarkably similar to enhancer sequences. And they each even have proteins that bind to the telomeres that are remarkably similar to what are called transcription factors, like TRF1 and TRF2 as two examples. But I believe that the telomeres are actually constantly folding over, coming in contact with genes like a magic wand, are turning genes on and off. But as the telomere gets shorter and shorter and shorter, it can't reach as far as it used to. And as a result, it changes the epigenetics of the genes. And that's why epigenetics is like a dimmer switch. Genes can be at all different levels, like a dimmer switch to turn lights off and on. So we have found, and others now have done this studies too, when you look at genes that are appearing to be regulated by telomeres, and you take a cell line where you can turn telomerase on and turn it off and turn it on, turn it by what's called inducible expression, you can lengthen and shorten the telomeres back and forth. Make the culture have long telomeres, then let them get short, then make them long again. And when you do that, you find that those gene expressions, the epigenetics of those genes that I just mentioned, they go up and down proportional with the telomere length. Okay. Now I have a theory, as I said, the folding over like an enhancer sequence. I don't know how to test that, and I haven't even tried to, but the fact that it does happen is a reality. And a lot of other theories have come aboard that try to explain why that is, but nobody really understands how that happens yet. And maybe we don't need to understand it. We just know for a fact that lengthening and shortening telomere, doing nothing else to the cell, affects the epigenetics of those genes proportional to the telomere length.
Okay, excellent. Yes, thank you. That makes sense. Okay, Dr. Andrews, thank you so much for joining us today. Where can people go to follow what you're doing? And you've written a couple of books.
I've published 2 books. One is called Curing Aging, and the other one is called Telomere Lengthening. I co-authored those with a guy named John Cornell, who is actually a really good writer, and I'm not. But you know, we self-published. The goal was to just provide an opportunity for people to get the answers to all the questions that everybody asked me. Okay, so the books are designed pretty much to talk about the basics, but we really haven't kept that up. I mean, Amazon, we were selling on Amazon. Amazon ran out a long time ago, and we haven't sent them, or we haven't had any new books made. So if people wanted a copy of the book, it was never to make money. Okay, it was just to educate people. If people want a copy of the books, they can email me, and I can send them a PDF of the books, of both books. And feel free to provide my email address in this video, but I'll just say it right now. It's B as in boy, Andrews at Sierra Sci, that's SIERRASCI.com. And it's really simple for me to just reply with the books attached to the email. But the better thing to do, in my opinion, is, if you're like me, I always prefer to watch the movie instead of read the book. But I do have some great videos, and they're pretty much in reverse chronological order. So if you go to my website and click on videos, you'll see the most recent one to the oldest ones. But there's some older ones that are actually pretty good. And one of them especially is called Everything About Telomeres. And it's a 2-hour video, which I would never watch a 2-hour video but it starts with a table of contents. So you can go to the table of contents at the beginning and figure out why are you watching the video? And because I answer every question you can ever imagine. Okay, and you can go to that time and then just watch the 5 minutes where I discuss, like why does the length of a telomere affect aging, things like that, so those are places people can go. And there's a lot of videos that have whole new subjects since that Everything About Telomere video, and those would be the more recent ones. And what I've done lately is I've kind of done a series of what I call the what, why, how, and how not of aging. Now, I can't talk about all 4 of those subjects in a reasonable time, so I've got the video on what aging is, and really it's just aging. All aging is a decline and failure of multiple tissues and organs. Nothing more than that. Okay, and so then the why we age, I have there's few videos, some ranging from 20 minutes, some to about 35 minutes, on why we age. And I go in great detail explaining evolution and how evolution, all through the time of evolution, it's been a trend towards increasing diversity. They can go watch those videos. Then I have videos on how we age. I talk about how tissues and organs decline, and how there's different types of cells in a tissue and organ. There's frontline cells and there's reserve cells, just like in a military battle. Okay, there's the front line where the soldiers are. And so when a soldier gets killed, somebody from the reserve comes along to replace that soldier. Well, all our tissues and organs have the same kind of thing, but the faster you kill off the front line, the faster you replace with reserve cells. And to replace with reserve cells, you need cell division, which causes telomere shortening. So that's the how, at least for humans, some human primates, dogs, cats, horses, sheep, and pigs and deer. Okay, now, so that's the how, but all of them, every aging process does end up resulting in the faster you have wear and tear of your cells, the faster you age. So even though they don't do it by telomere mechanisms, they do it by something else. But the big thing is decrease wear and tear, period. And then the how not. I talk about the how not. And I don't talk just about telomere shortening, because I clearly say in a lot of my videos, the war is not going to be won with just one battle. Okay. And so telomeres, I think the main reason why I'm focusing on telomeres right now, when I believe in all the theories of aging, is because, as I said before, it's the one absolute limit on our lifespan. And I say we will never cure aging unless, in addition to solving the telomere, we'll never cure aging unless, in addition to whatever else we do to cure aging, we also figure out a way to prevent telomere shortening. So since that is the limit, that's why I'm putting my focus on telomere shortening right now. But I don't believe that preventing telomere shortening is going to be all we need to do. I just know it's the only thing that I am 100% certain, and not just me. I mean, there's any scientist working in cells and petri dishes and labs know that telomeres shorten when they culture their cells, and they can only have a limited lifespan. Well, our lifespans are limited by the telomere shortening, so we have to solve that problem if we ever plan to live longer and healthy for more than 125 years. And so that's why I'm focusing on that. And I'm very grateful that other people like Aubrey de Grey and others are doing other things, because I think we're going to need more. But I don't know. We won't know until we find out. But yeah, so go to those videos and you can learn a lot about the what, why, how, and how not of aging.
Excellent. Thank you. We'll definitely link to your website and people can find the videos there. Okay, Dr. Andrews, thank you so much for joining us today.
Thank you. Look forward to watching the video myself.
Okay, excellent. Thank you.